Well, hello everyone. We're going to let some people continue to filter in, but I just want to thank everyone for coming. We have some really great speakers today, and we're going to cover a lot of ground and quality control. We're going to start off with Avi, but just a quick reminder, we do have a Zoom Q&A feature if you have any questions, and we'll have a short Q&A session at the end. So feel free to either pop your questions in the chat or save them for the end. And Avi, I'll let you take it away. We can also maybe give it another minute or two, just let some more people file in.
I'll just get my presentation up.
Perfect. And this will be recorded and posted to YouTube afterwards. So we'll send the link to everyone in case you want to review any slides or, dive in any further after.
Okay. So just double checking to make sure it's just sharing my window and that the video
No, looks
overlay
great.
from the gallery isn't showing up. Okay.
Looks perfect. Feel free to jump right in.
Cool. Well, thank you for having me. So I'm going to give a very brief description on some of the NAB or NA beer safety compliance and what we like to call the hurdle stack. So as a lot of you probably know, um, when it comes to non-alcoholic beer, the biggest concern is the lack of alcohol. As far as food safety concerns go, you by processing or developing a non-alcoholic beer recipe and making a new product, you are essentially removing intrinsic safety features that are built into beer itself, which is what made beer kind of like one of the safest things to drink for literally millennia. And we typically refer to as safety locks, hurdles, intrinsic factors. So you'll hear like a lot of different nomenclature about that. And one of the biggest things about non-alcoholic beer is that they tend to be higher in residual fermentables, as well as typically higher pHs by the end of fermentation, and the added risk of late addition bioloads. So this could be in the form of other flavoring agents, including fruit, dry hopping, etc. So in a brewing or in a brewing environment, what you want to do is you want to treat non-alcoholic beer as a food safety problem. So during any production process, you're going to have to add validated controls to replace these lost hurdles. So the key message here is that non-alcoholic beer is safe only when these fundamentals are in place and verified. So this is just a diagram that I made up, kind of like visualizing the pentagram of standard beer hurdles that potential pathogens or food spoilage organisms have to overcome to be able to ruin a product. So obviously we have our ethanol, we have our low residual sugar, so there's very low fermentable carbon or very low usable carbon under a low CO or under a low O2 environment, plus the added stress of having high CO2 and a low pH. All of these tend to work together to really kind of block out anything that could potentially go wrong with beer during your normal processing. In a non-alcoholic beer, though, you have what we call a reduced hurdle stack. And it's not so much that you are missing these components, in this case, ethanol and low residual fermentables. It's the fact that without these two additional hurdles, you're actually weakening the other three hurdles as well. So in these cases, they're less effective as a whole when you're missing part of that pentagram of hurdles. So what I would like to go over a little bit is more, not so much about the intricacies of what you should be doing to make safe beer. It's more about some of the processes that you can start to take to start recording or keeping track of and refining your internal processes. So food safety is a really important subject for me. I did a lot of work in food safety. My background is food science and technology. I did a lot of computational biology and complex fermentation microbiomes. So this is pretty near and dear to my heart. And I tend to work a lot with food safety aspects as well as research and development into new and improved handling processes and brewing strains for the brewing industry. So what I want to talk a little bit about is mostly nomenclature and things that should hopefully start to engage in discussions for how you approach new sanitation requirements for a brewery should you be starting a new NA line. So I'm hoping, really, I'd like to see some hands up or maybe some thumbs up in the chat about who has heard of HACCP and HARPC. And if your brewery has any of these plans already in place, be it just for normal beer production or for any other process that you might have when dealing with food. So take a moment, think about that, but we'll go over a little bit of the fundamentals. So with HACCP and HARPC, in plain terms, this is a way to essentially document all of the hazard analysis, preventative controls, CCPs or critical control points, their critical limits that essentially flip the circuit breaker for when you will have to take action, as well as monitoring the records and the corrective actions that need to take place within the scope of your HACCP plan, as well as verification and validation of those plans. So when you're creating a HACCP plan or an HARPC, you will want a written plan and records, because essentially if or when something does happen, it's pretty much a numbers game at that point. This plan is going to be your fallback. This is going to be how you are going to react to anything that could possibly go wrong during your production process. And so now a little bit about the production methods used for making non-alcoholic beers or very low alcoholic beers. These fall into three primary categories. We have our maltose negative yeast. So these ferment just simple sugars, usually just glucose and fructose, and leave maltose, maltotriose, and larger sugars untouched. So this produces low ethanol from a standard wort, usually from a diluted wort. And the risk point here is that now once you're done with fermentation with your maltose negative yeast, you have a ton of residual still fermentable sugars left over. This could leave you open for cold side contamination, and this makes things like filler hygiene really important. This also is very similar to arrested or restricted fermentations. So the main difference between a maltose negative yeast fermentation and an arrested or restricted fermentation is that with an arrested fermentation, you can use a normal Saccharomyces cerevisiae brewing strain, but you purposely limit the amount of time it can ferment or the contact time with the substrate, such as the wort. So a lot of these, you have the same kind of conditions that you want to watch out for. It's the same kind of hurdles that you'll have with the maltose negative yeast. You want tight control of pH, your IBUs, your dissolved oxygen, and your packaging hygiene. And then we also have your mechanical separation or your dealcoholization and back blending. So this makes use of warm surfaces to basically volatilize the ethanol under a vacuum. You reblend that in with maybe some of the RO water that has been infused with volatile organics that constitute the flavor. And then really here you have your post-process recontamination windows because you're dealing with essentially a warm product. And when you're diluting your product from a full strength product, you're also weakening a lot of those hurdles. So for understanding these fundamentals, particularly the hurdle stack, you need to really kind of look at how your beer or how your product is going to be formulated, regardless of whether or not it is beer. Under these conditions, particularly with like a maltose negative fermentation or an arrested fermentation, your target pH is going to be pretty high by the end of that fermentation. So you want to make sure that you're hitting but less than or equal to 4.5, pH 4.5. This is a classic low acid concept buffer range that we aim for because one, underneath or below pH of 4.8, 4.6, Clostridium botulinum cannot sporulate. So that's kind of considered the gold standard in food safety for a low acid product. These can be adjusted using phosphoric acid, lactic acid, any kind of food grade acid. And you want to use bitterness as a supporting antimicrobial, but you can't over rely on it. Mostly because the alpha acids are only really going to affect gram-positive bacteria, and it's typically going to be those associated with food spoilage rather than with human pathogens or foodborne illnesses. And you also have to account for late additions. So any kind of sugar that's added or part of a late addition or post-processing addition, such as a fruit flavoring, syrups, botanicals, and dry hopping. As part of the hurdle stack as well, you're also going to want to take into account the lethality. And in this case, when we're talking about lethality, we're talking about lethality to any spoilage organism or foodborne pathogen in the product and preventing any sort of enrichment. This is done through tunnel pasteurization. And this is what we consider the gold standard in the food industry. Any kind of pasteurization process is going to be the safest way that you can handle any kind of food for longer shelf life and overall food safety. It's a highly validated methodology that there's a ton of research on it. So it's pretty easy to find the information that you would need to really start to implement that in your own brewery or food manufacturing facility. But this still requires constant temperature distribution and heat penetration studies within your products, as well as routine monitoring to make sure that all of your products are treated the same way or that they're treated adequately. Still filtration, this is also gaining popularity. This is any filtration that's below 0.5 microns because this is specifically bacteria retentive. So below 0.5 microns, you should be able to get rid of the vast majority of problem-causing bacteria. And then 0.65 microns, that's only going to be for yeast. So that's more for like shelf life stability in a standard beer rather than spoilage in terms of like over fermentation. So if neither are feasible, you can route your non-alcoholic product through refrigerated only pipelines with a short shelf life and controlled distribution. So this needs to be stated plainly in your plan, as well as accounting for any sort of infrastructure that handles that. I highly recommend if you're going to go this route, like anything that has to do with a tunnel pasteurizer or sterile filtration, that is the best way to go. And then finally, as the third part of these fundamentals is the hygiene and oxygen management. So as we discussed earlier at the start of the presentation, that one of the primary hurdles is low O2. And one of the things that foodborne illnesses require, or any of the pathogens that cause those foodborne illnesses, is oxygen. And sometimes they don't really need a whole lot of oxygen. Most of these organisms tend to be obligate aerobes, but a little bit of oxygen can go a long way in terms of potentially setting up like a sort of like a subcritical baseline population. And you're going to want to be able to verify your CIPs and SIPs on particular hotspots. So these are like your filler valves, gaskets, heat exchanger plates, your RO, and your filtration membranes. You're going to need to minimize any sort of dissolved oxygen pickup after lethal or after your lethal step or filtration, and keep things cold because heat and oxygen will accelerate biological risks as well as flavor, as well as detriments to your flavor. So you're going to want to make sure that you're using closed transfers post lethality and to avoid any kind of open handling. So one of the things that I myself have been working on the most, at least the ones that I have the most experience with, are these maltose negative programs. And one of the things that we like to discuss with brewers that use maltose negative yeast is what are their critical and practical checkpoints that they have in their processes. People that are successful, that have developed high value brands based around this, tend to all have like the same sort of checkpoints. And these are the wort design. So you want to simplify the composition of your wort. Basically, you want to make it so most of the sugars that are left over are those that would be typically left over and unfermentable in a normal beer as well. So that means getting a very high fraction of glucose to your maltose and maltotriose. Well, anything higher than that, that's okay to have, because obviously that's going to do things like add mouthfeel, maybe help stabilize flavor, and aid in packaging and foam stability. pH management is critical. You want to confirm that you're below 4.5 after all additions and blendings, and packaging. So cans, tunnel pasteurization, 0.45 micron filtration, anything that you can use to essentially physically remove or physically deactivate any living organism that is in your product is going to go is the way to go. You're also going to want to make sure that you're working with ultra clean filling, and that your kegs are well maintained. So if you're not shelf stable, and this is a test that you can do in your brewery, basically monitoring any long-term or short-term changes in gravity, or pH, you're going to have to treat as refrigerated only and short dated. This is a question that we get asked a lot, at least in my line of work, is how to handle things like non-alcoholic beer on draft. And my general answer is this is something that you definitely need to proceed with caution. This requires a lot of planning to really ensure that what you're doing is going to protect not only your customers, but also your brands. Because the fact of the matter is the draft systems themselves, these are hosts to very complex and highly resilient microbial systems, mostly because they're exposed to the atmosphere almost all the time, like the heads are. And there's could be potentially be a lot of movement between the heads or the taps themselves and the lines. And this partly because what people oftentimes don't think about is the infrastructure itself. So if you have very long tap lines, those tend to be much harder to clean and clean effectively, mostly because like you have to ensure that you're cleaning all parts equally when you're doing your line cleaning. And if you have like, 100 meters of line leading from your bar to your basement, that's going to be virtually impossible with a system that that really only dispenses a highly nutritious, hurdle deficient drink product. So if you absolutely must pursue NA on draft, then you have to do it with a shelf stable product, and ensure that you have validated line cleaning methods involved, as well as commitments to maintaining that cleaning regimen. So you're going to want to prefer a strict cold chain and short dating system, and going to avoid long ambient distribution. So that's where you have lines that could potentially be exposed to just ambient temperatures for long periods of time. Basically anything where the liquid is sitting for too long, because microbial contamination can happen fast, especially at warmer temperatures. So the QA and QC aspects of it. So you have your minimum viable controls. These are things that you are going to want to check mark for each aspect of your plan, and that you're going to want to verify. So per batch, this is going to be something that you want to do on a per batch basis. You're going to want to ensure that you hit your final pH, that your final dissolved oxygen and your IBU limits are at your control point, as well as hygiene checks. So you're going to want to either do things like ATP swabs, plating, and checking of your filters for your deaerated water and RO loops. And these are things you're going to want to record, so you can always go back and check for trends and reviews and see like where things need to be improved. You also have to have a micro plan in place. This is something where you want to do your typical beer micro, as well as looking for potential contaminants. Now, if you're looking for food safety aspects, I do not recommend trying to enrich for food pathogens in your own lab. This is something that you can use a third party for, mostly because, like, one, most brewing laboratories are not equipped to be able to handle biosafety level 2, potentially biosafety level 2 hazards, and two, they often require specialized equipment to be able to properly incubate and specialized agars. And you're going to want to do a shelf life check. So this is a real time, so monitoring the shelf life in your typical storage solutions, as well as accelerated aging for pH drift, anything to do with gushing, bulging of cans, popping of bottles, off aromas, flavors, and what we would refer to as microbial outgrowth, or anything where you start to see blooms of microbes that shouldn't be there. And again, there should be no microbes blooming there in any NA product. So incident readiness. This is going to be a when, not an if, especially if you're in an environment where you're producing a lot of product. At this point, it is just a numbers game. So in your HACCP plan or your HARPC plan, what you're going to want is a specific section dedicated to a recall and incident plan. These will dictate important people. So these will dictate who has what roles and what they're supposed to do, after hour contacts, complaint intakes, what to do for escalation if these need to be, if you need to trigger a recall, or if you need to trigger a check, hold and release rules, regulator and retail communication templates. So these are all things that you need to have on hand because you want to do this as quickly as possible to avoid any potential damage to your brand and to customers. And then you want to run at least one mock recall annually. This is just to ensure that everybody knows where the plans are, how they're supposed to be interpreted, and where, and what to do. And you should be able to produce a physical copy of your HACCP or HARPC plan, monitoring logs, and validations on the same day that the complaint lands. Granted, you could do fine with a digital copy as well, as long as you know where those are and that the correct people have access to them. So these are going to be vitally important when thinking about setting up your own non-alcoholic beer system. So to finish off, there's definitely a lot to cover, and I really wish that I could spend hours going over this. But unfortunately, I'm going to give you just primarily the basics, and these slides will be made available. I passed them on to Max, who could pass them on to you if you're really interested. Because really what I want to do is just introduce the terminologies and a lot of the key things that you should be thinking about when thinking about food safety in a brewery. So the primary takeaways are the NA removes natural protections. When you have that pentagram of the intrinsic properties of beer and you remove one or two of them, you're not only removing potential hurdles, but you're also weakening the other hurdles as well. And then write it down. So if it's not written down, if it's not recorded, then it doesn't exist. And if it doesn't exist, that could potentially get you into trouble if you're audited. So you want a preventative control, you want a HACCP plan, or an HARPC. Sorry, that's what I wrote HACCP twice, with clear limits, clear monitoring situations, corrective actions, and verifications. And then really, for me personally, no NA on draft unless you have a shelf stable product that is verified and tightly controlled. Because the thing is you have to understand the risks of any kind of not intrinsically safe food being served in the way that draft beer is served. So with that, I will.
All right, Avi, thank you so much. And now we'll move on to our next presentation. We're having Wade Wade Begrow from Founders coming right up. So Wade, I'll have you share your screen and take it away.
All right, thanks, Anna. Let me get this pulled up here. Great. All right, I'm going to start my timer here. Thanks so much for having me. This is a topic that I could talk for days on, but it's a challenge to squeeze it all into 15 minutes. But today, my goal is to give you a brief overview of brewing microbiology, and then maybe give you some tips and tricks that might help you at your brewery. So to start, I like to use Slido for presentations. So if you don't mind, or if you'd like to do it, go ahead and scan this QR code, and then it will open up a web page. And I have a couple questions for you throughout this presentation to help me learn more about you. So, and there will be another QR code if you missed it. We're going to talk about introduction to brewing microbiology, major beer spoilers, what are some of the micro testing methods that are commonly used, and then again, recommendations and best practices for small breweries. So I have a question up right now, and if you need to rejoin, you can go ahead and scan that QR code. But in your opinion, why is brewing microbiology important? And these will just sort of pop up. Safety, love that. Product stability, yep. I'm really glad that I'm following the non-alcoholic beer talk. These are all very crucial things to look for. Flavor, sanitation, absolutely. Keep them coming. Consistency, yes. They're doing the work. Okay, great. I'm glad that somebody brought that up because, yeah, after all, if we didn't have microbes, it would be a dull, sober world, and I would not be at the brewery right now. I'd probably be at a dairy plant or something like that. So, yeah, we need microbes to make the alcohol. We can't do that as humans. We can't wave a magic wand and say make ethanol. No, we need yeast to do it. But also, yeah, there can be some serious problems that can come in if you don't have control over it. So, great feedback. Thank you for that. So let's talk about, yeah, what are the threats? I really liked how Avi talked about the intrinsic hurdles that are present in beer, and for the most part, this focus is going to be on full strength beer. We do make non-alcoholic beer at Founders. We're just starting to. We have to watch that very closely from a food safety and microbiological standpoint. We're really fortunate that we have a tunnel pasteurizer where we can send sealed cans through. And yeah, there have been asks to have it on draft in the tap room, but I'm not quite comfortable letting that happen yet. So these are the microbiological threats for full strength beer. And because of time, I really just want to focus on a couple major spoilers there. So Lactobacillus brevis, or Levi-Lactobacillus brevis, and Pediococcus. The names have changed a little bit in recent years, and I'm still kind of adapting to that, but we'll talk more about that later. And then Saccharomyces cerevisiae diastaticus. That is a major wild yeast spoiler that can cause serious damage to your brand and your product. So as I mentioned, we'll just focus on these two. Levi-Lactobacillus brevis is a gram-positive rod, and the really nasty strains have hop resistance or hop tolerance genes. So they're able to sort of get rid of those acids that normally would kill them, and they're able to thrive in that that beer environment, low pH, low oxygen, high alcohol, all those things. So keep that in mind. And what can happen? Well, if they get into your product and you don't want them there, they can produce lactic acid. They're lactic acid bacteria. They can also produce other organic acids. They'll drop the pH. They may cause your product to be turbid or even kind of ropey or sick beer, slimy beer. Definitely not good. So again, they're not pathogens. They're not going to kill you or the customer, but certainly are detrimental to to your product. So the name brevis, it comes from brevity or short. They can appear long if they're not adapted to beer, but generally they're short rods if you see them under the microscope in beer. Let's talk about arguably the most notable wild yeast, and that's Diastaticus. And by definition, as far as I know, it's a strain of Saccharomyces cerevisiae that contains that STA1 gene, and that encodes an extracellular glucoamylase, an enzyme that is secreted outside the cell that can continue to chew up some of those complex starches and liberate simple sugars that can feed that yeast and whatever else is in the package there. That can cause gushing. It can cause increased alcohol by volume, increased turbidity. Some may give a phenolic off flavor, so an off flavor that you don't want in your product if that's the style you're going for. And then the biggest risk, and arguably the biggest risk to the consumer, aside from drinking too much, is package failure. So when I say package failure, I mean cans and bottles exploding, that sort of thing. I just saw a recall for a 5-liter keg that was recalled because of secondary fermentation, and that certainly would make a big boom if it blew up. So I hope that I've made the point that these are dangerous. I would argue that both of them have a place in brewing for some styles, but we stay away from them here. So let's get back on your phone. Let's do another poll, and this will help me kind of gauge the audience. I looked at the attendee list, and I recognize some faces, some colleagues there. Hi, guys. But I don't know about most of you. So do you have a micro testing program? If not, that's okay. But if you do, kind of describe it and let me know what you have there. So I'll wait a little bit for people to answer. I think I set this one up to give the response once I click, so I'll give it a little bit. Okay. Oh, actually, here we go. Okay. Yes. Okay. So some people say sort of. I imagine some of my colleagues use a traditional or a combination of plating and advanced testing like PCR. Okay. That's good to know. How about the next one here? Do you work at a brewery? And then if so, how large is it, or how big is your annual production capacity? Let's see if I can get the results here. Oops. Okay. Okay. Yeah. So some medium-sized. I developed this talk to kind of focus on breweries producing 20,000 barrels or less, but this certainly could apply to anybody here. Awesome. Let's talk about some of the basic equipment for microbiology that I recommend, and some of the advanced products as well. And again, I really only have time to cover a few of these things, but basic things that you should have when starting a microbiology program: a pressure cooker, hot plate with stirrer. In fact, the hot plate, you can make a lot of media just with that, but the pressure cooker really helps you sterilize it. But media like HLP that we'll talk about on the next slides to come, it does not require autoclaving or a pressure cooker. So you can do a lot with just a hot plate and a stirrer. Microscope is crucial for not only counting cells for pitching, but also to look for spoilers and help identify what you see on those plates. Again, a pH meter is pretty crucial for brewing, but it can also help detect if you have a lactic acid bacteria problem. Clean space for preparing media. You don't necessarily need to have a laminar flow hood. That's what I recommend, and that's in the advanced column for a reason. But a lot of these microbes are not going to be like floating around in the air. They are beer spoilers. They like beer. A lot of them are strictly anaerobic, and it generally is okay if you have a clean space to perform the plating. Gram stain kit is good. Again, it can only tell you so much. One thing that I wanted to focus on is proper sample ports. If you're setting up a program and you're doing the work to collect the samples, you want to make sure that you have an appropriate sample port and you're not causing contamination simply by sampling. We really like the all stainless steel Perlick valves, so we autoclave them in between uses. Look for something that can really hold up to sterilization temperatures, and that will ensure that you're not having false positives from the act of sampling. Then also there are autoclavable keg taps too. Sterile sample bags or tubes, those are a no-brainer. You need something that's clean to collect the sample in. And then, as I mentioned, some of these microbes are anaerobes. They will die and not grow on a petri dish in the presence of oxygen. So if you're just starting out, I recommend these anaerobic generators. They're commercially called gas packs. There are some other versions out there, but they're like little hand warmers that help scrub and reduce the oxygen from inside that bag to create an anaerobic environment for them to grow and thrive. On the right side, we'll talk about some of the advanced equipment. I recommend an autoclave as soon as you can get one. And then we use real-time PCR or qPCR. That's what is kind of the gold standard right now, at least for identifying these spoilers. And then we'll talk also about how membrane filtration can really help improve your levels of detection too. I put ATP meter in advance, but I would argue that it could go in either column. It is a very powerful tool that you can use for real-time results of how clean your process is, but it's not the best at detecting really low levels of spoilers that could cause problems. So let's talk about some of the testing methods for these different microbes, and I'm not going to get into the details in all of them. There's just not enough time to talk about them, but I really like HLP, Sues lactobacillus, and Pediococcus media. And then WLD is also a powerful media that can be used to detect lactic acid bacteria like Levylactobacillus brevis. As I mentioned, PCR is the gold standard as far as I'm concerned for detecting and identifying these with confidence, but there are other genetic products out there, and I'm always up for learning more too. On the flip side, Diastaticus, it can be difficult to detect. It looks a lot like ale yeast. It is ale yeast, and so it can sort of hide with your house yeast strain. And I don't know anybody. I would never look at a cell under the microscope and say that's Diastaticus and that's not. It's just not possible. So the best way to do it is to use a selective media that will inhibit your house yeast and other microbes that you don't really care about so much to allow those wild yeast to grow. And generally, copper sulfate is used. So Lin's cupric sulfate media, LCSM, is a very popular petri dish that can be used. You can also use a broth with copper sulfate or some other compounds. The big challenge is sort of seeing through all the noise of the culture yeast and detecting that wild yeast in there. It might be at very, very low levels. Okay. I have some pictures, some show and tell. So this is HLP. It's a semi-solid media that goes in a tube. So it has about 2 grams per liter of agar, whereas full sort of regular petri dishes have 15 to 20. So it is like a gelatin. This is one that you can boil and dispense into tubes. Boiling drives off the oxygen, and you mix it. You can mix it with beer. You can put a loop in there. You can put a swab in there, but it's nice because you can visualize some of these spoilers as they grow. And lactobacillus generally has this sort of comet-like shape. Looks like they're going up, but from my understanding, they're actually going down. You can have a clear go, no go. These do take, a few days to develop. I would argue that they're useful, but maybe not as fast as PCR. But here you can see we've got growth in the center, and the two samples on either end are clean. Okay, HLP. Here's a picture of WLD, Wallerstein Laboratories differential medium, and this has a pH indicator. So if you have colonies that grow, that's one thing. But if you have acid production, they are going to change that color and sort of cause that halo there. So this can be a nice way to show acid production. And it also has cyclohexamide, which will suppress culture yeast too. As I mentioned, LCSM can be a powerful tool because it can suppress your house yeast. So in this picture, we have two different Petri dishes. The one on the left is WLN, so it's different than WLD, but I can't get into all the details today, but this will allow your house yeast to grow. That same sample that's plated on LCSM will suppress your house yeast, and this plate is clean. We can say that we did not detect any wild yeast. So how do we, get better and improve detection levels? I mentioned that. We use membrane filtration, and that is a very powerful tool. So we already heard about membranes a little bit, and this is generally a 0.45 micron membrane, sometimes 0.2, and you can put it on a vacuum filter system, filter beer down onto the plate. The microbes will be trapped on the top of this membrane. The liquid will travel through, and then you can, put this membrane onto a plate or in broth, and you can really increase your sensitivity. So, for example, if that last slide that had the clean plate there, if we put 0.2 of a mil on there, spread it around, we might have a clean bill of health there, but we might be missing low levels of spoilage. So less than 1 CFU per mil, or for one example, 40 CFUs per 100 mil. So you can imagine that you might miss that, or maybe you just see one colony and it's no big deal. It's just one colony. Well, if you filter 100 mils down onto a membrane, that's equivalent to doing the plating 500 times. So I wanted to make sure that I pointed this out. Membrane filtration is a powerful, powerful tool. This is the setup that we have in the lab. We have a vacuum pump and a trap and an autoclavable manifold. The whole thing can be autoclaved, and we can take these cups off, put the membrane on there, filter the beer down, and then we can put that membrane right onto a plate of LCSM and see what grows. Or in preparation for PCR, we can dunk the membranes into a liquid broth and then propagate the microbes. And hopefully we find a spoiler that way. Okay. So here's an example that I talked about. This is a real thing. I took this picture. This is very, very low levels of diastaticus, and I'm not going to count them now for you, but there are about 43 colonies on there, and you would miss it if you just plated it on a Petri dish. Here's one on the right that has higher levels, but again, even at 1,400 CFUs per 100 mil, that's 14 CFUs per mil. If you're only plating 0.2 of a mil, you might miss that too. So I don't want to downplay the importance of getting started and plating, but I do want to highlight that membrane filtration can really help improve your testing. Here's just another picture of that example. Just one colony, but if that one yeast cell is happy and is able to grow. It's just a matter of time before you could have some serious problems. Another tool that I like to use, these fermentation tubes, sometimes they're called like Fisher fermentation tubes, I think, but essentially it's a closed tube here with one opening. You can pour beer or whatever you want in there and flip it upside down to make the bubbles all come out. Then you can like inoculate it with a little bit of a colony, maybe of suspected Diastaticus, close it up with foil and wait. And here you can see this was just after a week or so. This spoilage yeast was happy. It made alcohol and CO2 that was collected in the top. It's a clear view of what happened. So as I mentioned, Diastaticus can secrete that extracellular glucoamylase and can turn dextrins into glucose and other simple sugars, which can lead to an increase in alcohol and CO2 and maybe package failure. We really like to use PCR. The tools that I showed you are handy, but really looking for genetic targets, that's the best way that I've found, at least. So this is a picture of our lab. We have 2 Roche light cyclers that we run on a daily basis. We don't just test beer, we test swabs and colonies and that sort of thing. But it gives us a clear go, no go readout, and can help us look at all of the testing results to make a decision on what to do. We use commercially available PCR kits, but we have also made some in-house kits. I recommend, if people are getting into this or are interested in PCR or some of the other options out there, just make sure that you select a kit that works on a variety of platforms. There are some products out there that really only work on one system, and you can sort of get yourself in trouble if you can't run it on a different system there. These are some of the common targets that are out there. So that STA1 gene for Diastaticus, there are a few hop tolerance genes that are also targeted. And then the list of major bacterial beer spoilers, there are sequences and targets out there too. So take a look at that, and maybe that will be something that you can bring into your brewing testing program. As I mentioned, we can test many different things. It's just looking for DNA and those targets. So you can pick a colony on a plate and you can run it. You can make a pellet from centrifuge beer, yeast slurry you can run. Again, it can be difficult if you have a lot of normal or house culture yeast cells and you have just a little bit of spoiler there. I recommend using broth to enrich the sample and really grow up or put the environment there to grow up that spoiler and suppress the good microbes. And again, if you really want to step your game up, you can start swabbing. So not just testing your beer or wort, but look around the brewery and find hot spots that may be harboring those beer spoilers. It's not always the case for us, but best practice is to wait to move the beer to a new tank or package the beer or ship the beer until the PCR results are ready. So time is super important. And oftentimes you're kind of going head to head with shipping and customers and when this beer is supposed to launch. So, work on testing, but also be mindful that you need to get results quickly too. So I know that I covered a lot, and maybe some of you are at different stages in your path or your journey. And I just wanted to end with this example roadmap of things or steps that I recommend. First, before you do any of the micro testing, I recommend just start tracking your batches and create like a warm storage of all the cans and bottles that you've produced. If you get a complaint from out in the market and you don't have any product to test, it's no good. And furthermore, if you are testing and you don't know what batch it goes to, it's just not worth it. So make sure that you do that to start. And then I always recommend starting a sensory program first. We can detect some of these off flavors, and you don't need to have any fancy tools and equipment to do that. Shelf life testing is important, especially if you're looking for some of these spoilers that might be at very, very low levels when you're packaging, but they can grow in, a warm closet or something like that. Also, make sure that you have a portal to record your customers the complaints that may come in. You may think that everybody loves your beer and it's problem free, but you'd be surprised if you have a place where people can submit complaints or give feedback, and that can help you track down problems. And then step 4, when you're ready, I recommend doing traditional micro testing. I would start with HLP and LCSM. I think those are great starts, but again, there isn't one auger or one broth that grows all of the spoilers at once. A lot of times they kind of compete and like different environments. So make sure that you select the media that works best. I recommend improving detection limits with growing things up in broth or using membrane filtration. And then, you can get into more advanced techniques like PCR, next-gen sequencing, that sort of thing, to do faster, more precise testing and more actionable data quickly. And then if you've done all that, I recommend that's when you sort of branch out and do more environmental monitoring or EM. Also, you can participate in proficiency testing. There are some proficiency testing schemes out there just for beer where they send you, an unknown can of beer, and there's a certain microbe in there at one concentration, and that can really help you too. And then, of course, what we're doing right now, and what I try to do, is to stay up to date with the latest methods and research, and just keep learning, because it's never dull. It's always changing, especially with the growth of non-alcoholic beer. There's a lot to learn. So with that, I want to thank you to the team that invited me, and thank all of you for tuning in, and thanks to Founders for letting me do this. So I just listed some of the resources that have really helped me. This QR code down here will bring you to the slides. So if you want to download them, you can go ahead and do that right now. I guess if I had to pick one or two things to do, I would order this Color Atlas and Handbook of Beverage Biology by Werner Beck, and it was published in German, but there is an English version out there. It's about $200, and it's not super easy to find. You have to buy it from the publisher, but it has a lot of things that we've talked about, but it has really great pictures of what you might see under the microscope and what to do about it. I have my Master Brewers hat on. Master Brewers is a great group of people that want to teach and learn and get better with making beer. So there are also some resources out there too. So take a look at these. Google Scholar can help you a lot too, if you just search, beer spoilers or that sort of thing. So I won't take any more time. I've gone way over. I just want to say thanks again for the opportunity to present.
All right, and with that, we'll move to our last presentation. We might run over a little bit. I understand people have to hop off, but Max, I'll let you take it away.
Yeah, of course. So first of all, thanks to Avi and Wade. That was a really great foundation, both for NA and for just the sort of fundamentals of doing micro testing for beer. I'm here today to talk about some emerging tools and sort of the importance of doing molecular testing, real-time PCR or just normal PCR, as a part of your process. And a lot of breweries, especially on the smaller side, as Wade mentioned, below 20,000 barrels, it can be difficult to do this sort of testing routinely. It's slow. Plating can take a very long time, and it's arduous to set up. Sending out to an external lab, you're waiting for it in the mail, and then you're waiting for the results. And a lot of times you want to get that product out quickly. It can be incredibly expensive. It could be up over $100 a test to send out to a lab for qPCR testing, and in-house systems can cost tens of thousands of dollars. Plus, you need to make sure you have trained staff in order to run it and all the auxiliary equipment. And a lot of brewers just don't think they need it. They would just rely on sensory. And especially if they've never dealt with a contamination event, it just doesn't seem worthwhile. But it's only when you kind of go through that experience that you really realize the importance of it. And a lot of things can also be invisible to culture. There are diastaticus can be difficult, as Wade mentioned, to look under a microscope and tell the difference. And certain bacteria can be very difficult to culture, or at least see. And that can mean contamination goes undetected, especially if you're not relying on these molecular techniques. If you have a contaminated batch, you're going to probably end up dumping that product, and that's money down the drain. But beyond that, there's damage to your brand if that product does go out, and there's costs related to recalling it, and there's a process, and it takes time. And all of that together comes to a contamination loss, right? It's not just the lost product and the direct cost of recalls. You might lose customers in the process, especially if there's off flavors or the beer's slimy. And that's really the biggest risk of these contamination events, is not necessarily the direct cost, but what it can do to your brand and just overall the amount of time it takes. And as I mentioned before, these threats can be hidden. There are viable but non-culturable bacteria where they're there, they're alive, but they're sort of sub-alive and they're not going to grow on plates like you would expect them. Diastaticus, as Wade mentioned, looks identical to brewing yeast under a microscope, and it's difficult to track down and it's difficult to resolve if you are dealing with diastaticus in your brewery. And there's all these environmental sources. I mean, all those raw materials are bringing in. That grain is just loaded with lactic acid bacteria, and it can live in hoses, in filler heads, and in biofilm between CIP cycles. And it can also be difficult to detect this contamination when it's at a very low level. And without some of these more advanced techniques, you might be missing things completely. And so that's why molecular testing is really important. You're detecting DNA from these organisms, and you're not relying on trying to grow them. And that is more so the gold standard, and you catch a lot more. And you're getting gene level intelligence. We talked about hop resistance genes, HORA, HORC, HIT-A, that allow these organisms to grow in these hop environments. And for diastaticus, the STA1 marker that leads to that potential gushing and over attenuation and things exploding. And speed really matters. If you're trying to get some value out of these results, you want them much faster. You don't want to wait days or weeks. You want to get your product out the door, and you want to know what's going on in your brewery. And so at Acorn, we developed a new system called Sentry, that's for detecting these brewing spoilers using molecular techniques, using real-time PCR. And we worked with the Technical University of Munich to develop our assay to test for 28 different beer spoilers, Lactobacillus pediococcus, Megasphaera pectinatus. These are the most common spoilers. Over 70% of spoilage events are going to be because of these bacteria. But also these hop resistance genes that allows that bacteria to grow, and 19 different isolated diastaticus yeasts from a variety of different sources, including brewery and elsewhere, so that we really wanted to cover all the diastaticus out there. And what we really worked on was the sample to result process. We try to simplify it as much as possible so you don't need that complicated lab setup and that lab staff. We like to say if you can follow a recipe, you can use this product. You collect your sample. It can be from your fermenter, your bright tank, your packaging line, or environmental swabs, as Wade mentioned. You enrich that sample. This is something we really recommend doing because it gets that detection limit down to the cells or cell-forming units per milliliters and not hundreds or thousands. And that process, you're just adding that sample to a broth that we provide. Um, we recommend 18 to 24 hours of enrichment time. That is hands-off time. So you can set up your process so that you're collecting samples and the next morning running your test. And then you extract that DNA. This is an area where the process is very simple compared to other techniques. We work with Lucigen Biosciences to get a really great extraction process that is fairly hands-off and simple to run. And that's 15 to 30 minutes of time. And then it's very simple. You put your sample into the provided tube, you put it in the machine, and you press start. And that device it can fit in your brewery. It's about the size of a growler. It really is easy to use and work into your process. And overall, we're trying to make this advanced testing more accessible. Avi and Wade are complete whizzes, but not every brewery can afford a lab technician. And so, like I said, if you can follow a recipe to make your beer, you can use this test. And we make sure that brewers have everything that they need literally end to end. Everything comes to you. So you're not sourcing extra reagents, dealing with multiple companies, trying to buy extra equipment. You have everything you need for the entire workflow, and it's turnkey. And like I mentioned, it's a compact benchtop device. You don't need, a complex lab setup. Like I said, it's the size of a growler, and it's built for breweries and how breweries actually operate. So you're getting the results on the device. You're not trying to deal with these analytical softwares, paying for licenses, or working with a third-party lab. That being said, we're always here for support, whether that's technical or to analyze results or just to help out. We work directly with brewers to make sure that they have everything they need. And so just as a case study, we worked with a brewery here in Illinois. They're around 5,000 barrels a year. They relied solely on sensory testing and, would occasionally send out, they had options to send out to a third-party lab, but very rarely did that. And they couldn't afford nor did they have the staff to use systems that are currently on the market. And in our short pilot with them, we detected spoilage in 2 barrels prior to their canning, which all in all was tens of thousands of dollars in recall costs and in reputation damage. And we achieved over 98% accuracy and going up as we continue to work with them, reduced quality control turnaround by over 75%. And like I said, estimated tens of thousands of dollars in loss prevented. Because they didn't have to deal with recalling and were able to track down and stop and not have any contamination since then, which is great. So as you're building your program, we really recommend having on-site molecular testing for routine screening. You can test every batch, you can test the environment. It's a great go, no-go before moving on to packaging and distribution. And plating and culturing is important, and it's great to follow up on positives and to understand the viability of the organisms that you're dealing with. And then, of course, external labs doing things like NGS, I really doubt many breweries, especially on the smaller end, can set up a next-gen sequencing program to do, but if you can, that is awesome. But those services are available if you're dealing with a complex or persistent problem. And here at Acorn, to touch on Avi's point, we're in mid-development for a panel to detect those pathogens, specifically in non-alcoholic beer. And we're working to continue to expand the coverage of our product, as well as the accessibility and ease of use, and driving down the cost so that the actual hardware and the reagents and everything is affordable to brewery, even on the sort of micro to nano brewery size. And then also continuing to integrate our workflow with how breweries actually operate in production records and expanding our software. So that is just a quick overview. We are proud to be partnered with the Independent Brewers Alliance and the amazing folks over there. So if you are an IBA member, definitely reach out to them or us if you're interested in the specific member benefits that we have with our program. We're also available through the Beverage Federation as well. And of course, you can reach out directly to me. My email's there, as well as our website and the specific brewing page. And in terms of questions, I tried to address some of them that came up, but we are I think already 4 minutes over. So feel free to follow up with me, or of course we can connect you with Wade or Avi if you have persisting questions. But we'll make these slides available and a video available for you as well.
Thank you, everyone, for coming. This is great. And feel free to send us an email either at anna@acorngenetics.com or max@acorngenetics.com. And we'll be sure to either connect you to the right person or answer your questions there. Thank you all.